The experimental station La Mayora is a singular facility unique in Spain and through Europe, dedicated to research into subtropical fruit trees. La Mayora belongs to the Subtropical and Mediterranean Hortofruticulture Institute, a joint institution between the Spanish Council for Scientific Research and Malaga University. La Mayora has an extension of 51 hectares with field plots and laboratories and specialized staff to carry out all kinds of research related to subtropical fruit trees and vegetable crops adapted to the Mediterranean area. Its relationship with the agriculture productive sector has marked its research lines since the 70s, when it began to promote strawberry production under plastic greenhouses. Its location in a microclimatic area suitable for such crops and in agricultural environment which favors a close collaboration with the productive sector are features which make this station unique. At La Mayora, research is aimed at fostering sustainable and environmentally friendly agriculture, trying to solve problems which affect important crops of the Mediterranean basin, especially of southern Spain. Physiological problems related to disease and pests are dealt with from different approaches. Regarding greenhouse crops, one of the main problems nowadays is related to the quality and quantity of available water for irrigation. Water salinity due to the proximity of the sea and the features of Mediterranean climate produce a low efficiency water use in the crops, which leads to important losses of production and crop quality. The plant breeding group for the greenhouse tomato crop production is involved in studies of genetic and physiological bases determining the efficiency of water use in plants. Root architecture and anatomy are being studied in relation with plant water uptake and plant water status. This valuable new information will help to support the development of root-targeted breeding strategies to generate new tomato varieties with increasing water use efficiency under the high evaporative demands usual inside the Mediterranean greenhouses. La Mayora's facilities allow precise physiological studies in growth chambers with a close of environmental growth conditions and nutrient supplies. We can see on this video the equipment used to register the photosynthesis and plant transpiration. The station has a seed bank collection with more than 1,200 accessions of tomato and 900 of melon. In this germ plants bank, a great part of the genetic variability of this species can be found as it conserves and treasures heirloom, traditional and local varieties that retain the good flavor of the fruits, and it also keeps improved varieties and wild-related species which have natural genetic resistance to pests and diseases. Research on tomato breeding is mainly based on the genetic variability that exists in this germ plasm bank. Interesting genotypes with valuable agronomic features related to pest and disease resistances or to the quality of the fruit are studied in order to discover how these characters are inherited. In this way, resistant commercial varieties with tasty and attractive fruits can be obtained. The use of wild species relative to the cultivated one is crucial for tomato genetic improvement. Most of these wild species collected in origin in Peru have little or no fruit quality, but contain valuable genes to confer natural resistance to viruses such as tomato yellow leaf spot virus, genes that can be transferred to commercial varieties by crossing and selection breeding methods. In this way, the research group strives for genetic improvement to solve problems such as the low organoleptic quality of today's commercial varieties of tomatoes or physiological disorders such as fruit cracking, besides those related to the incidence of pests and diseases. The most important research line on melons is related to resistance to diseases and pests. We work on melon breeding resistances to common diseases as powdery mildew always present in melon crops, and several viruses transmitted by aphids and whiteflies. In these images, we can see a plant with natural resistance to powdery mildew. These studies are carried out using both traditional breeding and molecular techniques. The obtention of resistant genotypes is carried out by using manual pollination. The introduction of resistance to aphids and whiteflies into melon crops is really important to control not only these insects, but the viruses they transmit when there is not enough virus resistance in this species. The picture shows some of the great variety of melons maintaining the germ plasma bank adscribed to the breeding department. This variability is very useful for melon breeding. Together with the study of resistant genetics, our group is also involved in the characterization of the found resistances in order to know their expression mechanisms and to identify linked molecular markers that allow a quick and efficient selection of the genotypes of interest in the breeding process. Here we can see how the fruits are analyzed. Plants and fruits are characterized and seeds are collected for the germ plants bank, maintaining the original variability of the melon accessions. The main objective of the physiology of development and selection of subtropical fruit germ plants group is the search for solutions to problems that arise when optimizing sustainable production in subtropical fruit trees. The different germ plants collections are characterized using both phenotypic characters and molecular techniques. With the DNA sequencer, the PCR amplified DNA of different genotypes is analyzed. Each variety shows different peaks at each locus, representing different alleles. The information generated is used both for selecting the best varieties and to optimize ongoing breeding programs. We have currently a germ plants collection of more than 300 Chirimoya accessions. The reproductive biology and the factors affecting fertilization and fruit set are studied to optimize production and yield. Studies on flower development and pollination, as we can see in these images, in the flower of an avocado, as well as pollen-pistil interaction, allow understanding the bottlenecks that limit the reproductive process. Fieldwork is combined with molecular techniques and microscopy. Methods of regeneration and genetic transformation are developed for functional genomic studies. The physiological basis of in vitro regeneration processes are studied to improve the efficiency of the transformation process in different fruit species. The optimization of the transformation process is necessary to carry out functional genomic studies. All these studies are performed in subtropical fruit species, mainly avocado, chirimoya, and mango, but work also involves other species such as lychee, carambola, and longan. In addition to all these research objectives, this group has also a transversal objectives based on a wide experience on production techniques in subtropical fruits. This experience is currently focused on the optimization of an environmentally friendly production. This transversal objective is the basis for the other goals and has a special relevance for dissemination to the productive sector in Spain and other countries. One of the lines of research of the horticulture department is the biological control of pests. Biological pest control is an environmentally friendly method that entails no risk for human health because it uses natural enemies instead of pesticides to fight against pests. At present, we are working with predatory mites from the family Phytoceae to control a pest that infects avocado trees, the Persea mite. After identifying the species of predatory mites and developing rearing methods in the laboratory, we are designing strategies to increase their numbers in the field to reduce pest populations and the damage in avocados. Another line of research aims at evaluating the effect of extreme environmental conditions, such as those of summer, on species interactions and therefore on the dynamics and the structure of agriculture communities. This research eventually aims at developing solutions to minimize the negative effects that global warming will have on agriculture communities that are managed with biological and conservational control strategies. The Plant Pathogen Interaction Group operates within the Crop Protection Department. This group works with pathogens which damage crops, such as viruses, fungi or bacteria. The group's aim is to have a deeper knowledge about the pathogen and its interaction with the plant in order to design more sustainable management strategies compatible with the environment and human health. A multidisciplinary approach is carried out involving laboratory activities using advanced molecular techniques for virus purification and nucleic acid analysis such as electrophoresis, genome sequencing, and sequence analysis. Field studies as well as activities in the greenhouse and growth chambers with environmental control are carried out. As an example, the tomato yellow leaf curl disease, which causes severe damage to crops and huge economic losses all over the Mediterranean basin and in tropical and subtropical regions of the world, limiting tomato production. Research into viruses associated with this disease is essential to design durable control strategies. In addition, a close collaboration with teams from the plant breeding department of the Institute has enabled the location of resistance to pathogens, which has been useful for control programs. At La Mayora, one of the research lines focuses on characterizing at molecular level the strategies used by several fungal pathogens to colonize the host plant and cause disease. These studies are performed using both a fungal species considered a model for molecular studies among the plant pathogens, Ustilagomydes, and a pathogen of agronomic importance, Verticillium dahliae, causal agent of verticillium wilt. Verticillium wilt affects a wide range of important crops worldwide. In Spain, this disease is considered a serious threat to olive production. Verticillium wilt also affects vegetable crops in Spain, sometimes with very severe attacks that cause economic losses. Recently, we isolated from mango trees exhibiting symptoms of verticillium wilt in the subtropical fruit-producing area of the province of Malaga. The long-term goal of these studies is to contribute to the development of effective and sustainable control strategies against agronomically important diseases such as verticillium wilt. Another line of research in mycology focuses on powdery mildew of cucurbits caused by Podosfera fusca. We study its epidemiology and its biological control, defining the diversity of populations of this fungus in the main producing areas of Spain according to different phenotypic characteristics. Most of this work is related to fungicide resistance. Maps of resistance to different active substances are established for the different growing areas. The resistant mechanisms to the different active substances are also determined, and the different capacity of adaptation of the sensitive and resistant strains are characterized. The action of various microorganisms to control the disease is also studied. The group also maintains a close collaboration with private companies through research agreements and technology support contracts, which eventually allows the transfer to the productive sector of the knowledge acquired through research. Summing up, the experimental station La Mayora is aware of the importance of applied research and interest in the transfer of knowledge to society for the improvement of agriculture processes so as to guarantee more sustainable and environmental-friendly production.